This is our second ever R&D update. The purpose of today is for us to be able to explain to you what it is we do, how it is we do it, and the progress we're making with the product pipeline. And today I'm very happy to have a very distinguished panel of speakers, as well as Antony Odell, our Chief Executive. The thing I really like about it is it brings home to everybody, you know, the real impact this technology can have on real people.
For those of you who are new to Tissue Regenix, just a short introduction to the company. Our business model is again a simple one. Tissue Regenix commercialises technology, that's our job. Money invested in tissue doesn't go into research. We work with our research partners, Leeds, Brazil and NHS Blood and Transplant, who are the experts in this space. That's their jobs, that's their rationale for being there. So we work with them to commercialise the IP rights that they generate, and we're going to be talking about the technology and how it's differentiated from other types of these approaches. We're very much a platform technology with a reduced risk profile, and that's basically because we already have clinical data that demonstrates that these things work. Our low-cost R&D strategy is also important. You know, we get lumped in with a biotech group in the London Stock Exchange, but we're not a biotech company, we're a medical device company. As a consequence, our timelines and costs are much lower. Essentially, we have one core patent which covers the process of decellularization of matrices. That's granted in the EU, US, Australia and Canada, and from that we file individual patents that cover specific products. As we develop the process for a specific tissue construct, we actually develop IP to protect that in the marketplace. We're making use of natural systems to do that. It's not an artificial thing, it's a natural healing mechanism that's kicking in here. Many decellularized tissue focus on the whole DNA immunology side. We focus not only on the immunology side, but also on the biomechanical side, and that's an important part. The patent that we have covers this low concentration SDS use, which is a detergent. We don't use chemical fixatives, and it gives a low-cost regenerative solution because I can order the stuff I need to make this thing off the internet. I don't have to go to Switzerland to have something specially designed. So in terms of capital expenditure, it's a very low hitter in that respect. Our programs are now being segmented into these 4 spaces: cardiac, advanced wound care, orthopedics and vascular.
Good morning, everybody. I've been invited to come and speak to you today about the biological scaffolds, really an overview of the development of the technology. Professor Fisher and I recognised the strategic opportunity of developing biological scaffolds for tissue repair and regeneration. We have evidence that they are regenerated in vivo by the recipient's endogenous cells. And we believe that the secret here is due to the scaffold structure and architecture. I'd just like to tell you quickly about 2 of the areas that we've been researching in. One is the development of arteriovenous grafts with reduced thrombogenicity. And 2 is the further development of surgical solutions for the replacement of tissues in the knee joint. We have been developing a meniscal repair device. What we're doing now is beginning to develop composite bone soft tissue scaffolds. So this can be used as a full meniscal replacement where the fixation into the knee joint is bone on bone.
And if the scaffolds work as well in the knee joint as they appear to do cardiac valves and dermis, then I think it will be extremely exciting.
Clinical applications of the decellularized valve allografts you can use for RVOT, right ventricular outflow tract reconstruction during the ROS operation, RVOT reconstruction for many congenital heart defects, for some monocusp repair during, for example, tetralogy of Fallot repair, and also for aortic valve replacement in humans. We published a tissue antigens journal that when we implant those valves in humans, there is no immunological reaction as you see in the cryopreserved. So you can see that donor-specific antibodies is much less on the decellularized when compared with the cryopreserved allografts. And this is true both for class 1 and class 2 antigens. With the decellularized allografts, we could eliminate some risk factors for late stenosis. In which patients those stenoses occur more frequently? In young patients, and you can see that for every age, the decellularized valves performs better than the cryopreserved. Also, some surgeons would match ABO compatibility between the donor and the recipient, and with the decellularized there is no need, because ABO incompatibility was not a risk factor any longer with the decellularized allografts. And also the size of the allografts that you need to use, normally you need to use an oversized allograft, which is not always available. And with the decellularized allograft, you don't need to do that, because sizing is no longer also a risk factor. Although cryopreserved allografts are still the gold standard, the incidence of elevated peak gradient is not negligible, and the use of decellularized tissue seems to be providing better reconstructions on those patients. So in our experience, the SDS decellularized allografts were superior to cryopreserved with less immunogenic reaction, lower gradients, no calcification, and partial repopulation of those grafts. So to sum up, I mean, our experience with D-cell valves, both on the right and in the left side, has been very gratifying, with excellent results and very low incidence of complications.
Heart valve, if you like, is the kind of ultimate thing, because it's being biomechanical, it's being vascular, and it's being, you know, all kinds of stuff. You know, vascular is a challenging environment from the point that you're contact with blood. Orthopedics is challenging from a biomechanical perspective. So, you know, there are very different challenges in all these environments, but what you're beginning to see is us responding to those challenges. You know, I think that's the exciting bit about tissue at the moment is, you know, not being, you know, Captain Kirk about it. We are going into territories we haven't been before, and we're showing it works.
The quality of the tissue that we could obtain with tissue Regenix technology, I mean, it's perfect for what we want, just to have a patch which will be completely repopulated by the host.
When surgeons open up an artery and then stitch it closed, we have the potential to narrow it and recreate the blockage very much for which we are there. If we are there to open up a blockage, the last thing we want is the body to then scar it down and make another blockage. That just is not a good thing to do. And so when we operate on blood vessels, a great majority of these times, we often put in a patch to cover where we're working. The ideal patch is something that is stable, that is durable for the long term. It has a low risk of restenosis and making another blockage. It has compliance near the host artery, and so it has long-term stability. It's very comfortable to sew in the operating room, and ideally for my trainees. It's easy to pull right off the shelf. This is why pericardial patches are becoming so of such interest, because they're not associated with bleeding, with infection, with pseudoaneurysms, and they are associated with long-term durability and stability. Now, the two most common options that one has are, one, either the pericardium, the lining of the heart, comes from either a cow or a pig. It's either bovine or porcine. In our country, we have bovine patches. Here in the UK, you have porcine patches, but they generally are thought to be really similar. I personally, as a surgeon, use pericardium. I use these patches in preference to the PTFE or Dacron patches, and I use them for multiple reasons. First, it is established for over 10 years. There's a nice size available, it comes right out of the package, and there's no report of any viruses, bovine viruses, human viruses, anything. They're safe, they're established. They're biocompatible. They've been, we believe that there's good tissue ingrowth, and that there's anecdotal reports. We can even use these in an infected field. Unlike Dacron and PTFE, which are filled with air, and are impossible to ultrasound, to duplex through early to see if there's a technical error, these are not. These one is able to investigate with a duplex, even intraoperatively, which many surgeons like to do. By 30 days, these things heal. You can see that they have a nice layer of neointima on them, which, looking at the neointima is composed of arterial stem cells. So what does this mean? This means that cells infiltrate into the patch, both macrophages and arterial stem cells. The endothelialization of the patch is complete by 1 week. And arterial stem cells are present in that endothelial position, and there's a complete neointima by 30 days and continued stem cell presence. The ramifications are for this is that in some patients who might be infectious, prone to infections, maybe they're diabetic or whatnot, that might be a group of patients that might benefit from this patch with reduced resistance to infection. I'd like to say that what I learned from these patches is that the tissue Regenix patches, they heal by stem cell infiltration. This promotes endothelialization and normal healing. It promotes resistance to infection. And I have to say, my impression is it's the closest thing on the market to a tissue engineered patch. It's the next technology. Other companies are trying to attract stem cells. They've already got the stem cells that are coming into their patch.
These technologies could completely change the use of prosthetics grafts as a surgeon uses for their patient. This is a remarkable advance. I'm very, very excited and look forward to hearing about more products coming through that I can test.
As a clinician, I'm dying to use this because I'm going to be able to tell my patients, I can tell my patients, your body will heal this. It will heal it because I know the stem cells are coming in.
I think that this technology is phenomenally interesting. We only studied it in the vascular system, so I can say in the vascular system it heals. But now there's the opportunity to use it in so many other places.
Human acellular dermis market from some recent research is $774 million. That covers a range of different applications, but it's a very vibrant market in the US. LifeCell hold a 49% share of that market, so they're very much the dominant partner in there with their alloderm and graft jacket. On the dermis side, in terms of development, on the human side, this product, as we know, was developed and validated by NHSBT. Let's look at the trial. All ulcers reduced in surface area by an average of 49.51% and volume 6 weeks after DCD application. And to date, we've got 9 patients completely healed. I think the important part about this study as well, and you'll see this when we come to do the full data set in December, January time, is we haven't just looked at this whole surface area business. You know, there have been a lot of studies where they're saying, oh, how big's the wound? Is it healing? Is it doing all this? We're actually looking at, are there vessels growing? What's the depth of the wound? We're looking at the volume of the wound. This is new technology. One of them happens to be an IP group technology called Icona. But this means that we're taking a far more scientific approach to this than has been done historically. The D-cell dermis has shown promising early results. 9 fully healed wounds. We're seeing more mature epidermis. One of the things we're doing in this study and people who are enrolled in it, we take biopsies from the actual dermis site. So we're actually looking at what's happening with the dermis structure as it heals. It's a very, very simple technique. Essentially, that ulcer there gets cleaned up. We use a Smith & Nephew Versajet in the trial. The dermis is laid across it, and we use Dermabond surgical glue to stick it into place. And then you put the bandage over the top of it. So it's really, really simple. And one of the things Ardy's most excited about this is it potentially using outpatients. You don't need surgery, you don't need anesthesia, any of these things. So from our point of view, it could move almost into a GP setting, that type of treatment. So, it's incredibly simple to use. And most of these patients have had one application. So from our perspective, we've got a very full pipeline, a lot to do. But at the end of the day, we're not having to reinvent things every time we do this. The process is fairly well fixed. It's a preclinical, clinical route. So this is not like stem cells where we're guessing at the regulatory pathways. We actually have a strong path to follow.
This is a technology that's completely leapfrogged conventional stem cell delivery because it recruits the body's own stem cells to this site. That's a tremendous advantage because the harvesting, processing, shipping, delivery doesn't exist. The body's done that for us.
Biological patches, the vascular patch market, biological patch market is fairly small, $34 million. It's dominated by synthetics, and probably the ratio to synthetics to biologics is probably like maybe 4 or 5 to 1 potentially. 90% of these are carotid surgeries, so it's this type of surgery for the biological patches. The other market opportunity within the vascular sector is the AV graft. Now these are Shire's figures from their recent purchase of Pervasis, but there are 820,000 end-stage renal dialysis patients annually in the US and Europe, and 70% of those receive dialysis. And these are the types of patients that would receive AV grafts, and AV grafts are essentially a way of preserving the vein through repeated dialysis. Human heart valves have been developed by our Brazilian partners in Curitiba. We have clinical data on pulmonary and aortic valves. We're in licensing discussions with a number of UK, German and other tissue banks, and they're keen to use the technology, and we're working out ways how they might do that. If you look at where heart valves were typically used, it's in the 60-plus patient group. But if you look actually back through the demographic, you'll see people are needing treatment for heart valve disease much earlier in that timeline. And current valves are not really indicated for use in that less than 60 years old group. So we believe there's a real opportunity for our products, which are going to be biologically derived products and decellularized products in that younger age group. Translation of the pulmonary system valve process is near completion, so that's transfer from Leeds into Tissue Regenix. And we continue our development on the aortic valve process. On the bioprosthetic side, we're working with our Brazilian partners to incorporate some enhancements into the design of the successful premium valve, which will incorporate decel technology, and we'll begin testing that in 2013. On the cardiac patch side, we have completed a pilot which was completed successfully in mitral valve repair and pericardial patching, and we have an atrial septal defect pilot in progress as well. Again, from research we've recently had done, we know there are 870,000 meniscal repair procedures performed in the US in 2011. Growth, not surprisingly, driven by rising obesity rates, and sports injuries are the two big drivers there. The ligament market, again a substantial market, dominated by synthetics at the moment and a little bit of allograft material, valued at around 528 million. So again, substantial opportunities for TR. The major point here is we have significant cash on deposit to drive forward our programs, and that's the important part. So from our point of view, we've got plenty of money to do what we need to do, and we've now got the team in place. We've got 99% of our recruitment completed. We've now got the team in place to start delivering that.
I think I'd just like to close it by saying that what you've heard today is some of the background into the research work that's gone on behind all this, and some of the pipeline of stuff which is yet to reach Tissue Regenix. We've also heard from practitioners that have been using this in surgeries. A lot of products coming through in 2013 to preclinical and clinical trials. And I know every company like ours would say that this year is going to be a transformational year, but I genuinely believe that 2013 will see a great deal of progress, and I'm certainly looking forward to standing here again next year and telling you just how much progress we've made. And finally, if we go back to that very first video, just remind yourselves about the impact this was having on those patients that have had those wounds for a very long time. This is dealing with real issues with real patients, and those issues are costing health services throughout the world an awfully large amount of money. So I think there's a compelling case, not just from the science, not just from the practitioner point of view, but also from the health economics point of view. And that's one of the things that we have to work more on as we go forward and nearer to commercialisation.